Okay. There you are. Now everything is
Yeah.
perfect. So sorry about that.
Okay. No problem. Yeah.
I'm glad we started a few minutes before, and thanks to our fans in the audience, they realized, oh, it's not recording. So thank you so much, everyone. Okay, I'll in the meantime, if I'm a little bit quiet, it's because I will put up the link again and share it on Twitter and so on. So I'll be back in a minute. Hi, Ilya. Thank you so much. You were the person that pointed it out. I really appreciate it. Thank you. Hi, Frank. How are you? Come up. Hi, James. Hi, Molly. Um, how are you, Frank? Haven't seen you in a while. Hope you're doing well.
Hi, Katerina. Yeah, it's been a little while and not that long. Yeah, so I'm doing all right and making progress along my own work as well. It's now I'm moving from mechanical and towards electronic simulations. Quite fun stuff. Very captivating with a little graphene sheet type of models. Yeah, really looking forward to this talk. Very interesting. Yeah, thanks for Ram to grace us with uh, your great research.
It's actually a very humbling topic. The more you work on it, you are just in awe with what nature has done.
Yeah, it's really a hard topic. I always yeah, look forward to these talks about those type of topics, but I also, it must be very humbling doing the research, but it's also, one of the most curious things to be working on, so it
Yep.
must be also really be very interesting.
Agreed. Agreed.
Oh, hi, Ram. Do you have a slides? What would be the style? I'm actually looking up the
Okay.
reference link
So I think,
Katerina
yeah,
posted.
yeah, I think Katrina will be sharing a link to the Google Drive where you will see the file, and it has animation. So it's not, when you click, it's not just going to go to the next slide. It'll show up pictures in a sequence, one after the other, so that the slide is not crowded. So if I show the slide in its entirety from the beginning, it's going to just show the, so if I click on it, you will see the slide. You'll just see the problems, origins of life. And then, um, so if you click on it, you are going to see this kind of a animation come up. That's what is going to happen. And so I'll walk you through the animation for that one. Okay, so let me know when I should start, Katrina, and I'll wait for your cue.
Yes, yeah, thank you for pointing that out. I'm posting in the chat if people come later to see the animation the right way. If you click on the link, use Google Slides. If you have the app, if you come here often to this club, it's really helpful to have it, since we usually share the slides through Google Drive. And if you then click on the presentation mode, so on the little play button on the top, then you can see the slides in the presentation modes. It's like a fuller screen. You can see it better, and you have the interactive videos and so on. So yeah, that's very helpful. Okay, I shared that in the chat, and we can, let's start. So welcome everyone to Science Society, and please, if you think this room is interesting for people you know and that are interested in this sort of topic, share the room with them. It's a public room, everyone can come and access it. And of course, a special welcome to Dr. Krishnamurthy. And before we start, let me give everyone a short introduction. And then after the introduction, Victoria will ask a few interview questions, since I think, talking about you in an interactive way is way more interesting than me just summarizing everything. So, Dr. Krishnamurthy, he did his bachelor's at the University of Madras and his master's at the IIT in Bombay. And then he moved for his PhD in chemistry to the Ohio State University, Columbus, and he there started actually to go on this path of science and asking the big questions in science, which is really interesting. The life or the path you took and the questions you had during this path of becoming then the professor at the Department of Chemistry at the Scripps Research Institute, where you are now. And so that we learn from it, I think it's a way more fun way if Victoria asks you interview questions about that. And thank you so much again.
Yeah, thanks again. Yeah, sure.
Thank you, Katarina, and welcome, Dr. Krishnamurthy. We're really so honored to have you here and hear you share your work. And so, yes, we would love to hear some background information that has to do, well, you're talking about the origins of life, and my question is about the origins of your interest in science. If you can think back through your life of when that origin arose for you, if it was in childhood or in school, or when you first really noticed that affinity for science.
That's a that's a very loaded question, an interesting question, because I have to reach very far back, because I come from a very philosophical background where we are generally the Indian philosophy is to question quite a lot. Why, and how, and what we see real, not to just accept what we see as real, but to keep on asking the questions and removing, the answer is not it is this, but the answer is not this. And that's a very strange answer. When we try to find out what the absolute real is, the answer keeps coming when we study this world as not this, not this, not this. And so it's that kind of a questioning which really set the stage for me. And I was very much interested in chemistry from a very young age, but then the spark actually came when I was watching Carl Sagan's Cosmos. And that was the first time it was being shown in India, and we would be in rapt attention every Sunday. It would come, and it would be wonderful to go on this journey with Carl Sagan on this voyager of his to the different planets and different worlds, and think about the origin. And that's where I started really thinking about why things are the way they are. It was really not an origins of life question, but why are things the way they are and not different? Why are they not otherwise? And those kind of questions kept on bugging me. And chemistry was the one that really kept my interest in science, because I was a very much interested in how molecules come together, how they give rise to properties that are, almost magical. How do these small atoms comes to become molecules, and they again give these supramolecular structures, and then suddenly all of a sudden you have a property that you can hold in your hand or be affected by. And then when I was doing my PhD at Ohio State, that's when I first came across this lecture by Albert Eschenmoser, who said, why not hexose nucleic acids? So that was a very funny question, and because when you look at your RNA and DNA, they are made up of what is known as ribose nucleic acids and deoxyribonucleic acids, which are 5 carbon atoms. But if you stop somebody on the street or even ask yourself, what is the first thing that comes to your mind when you say sugars? People will say glucose, fructose, sucrose. Rarely will anybody say ribose. People don't even think ribose as a sugar anymore. It's part of RNA. And so the question was, how did that nondescript ribose end up being part of the most important molecule for life, encoding information and having catalytic activities? So his question was, why not hexose nucleic acids? And that lecture completely bowled me over. After listening to the lecture, I simply fell in love with that subject. So that's how my interest started from a young age to this journey that now I'm still on. I would say the journey is not yet done, and I do not know whether it will be done even in many lifetimes. If trying to understand the origins of life.
Thank you very much. That's it. Yeah, I really resonated with you hearing you use the word magic, because that's really, I think for many of us, that's why we're here, and that's what we feel about science, that we're constantly delving in magic. It's amazing what we learn, and it's also amazing the questions that arise in us. And
Yep.
so, yeah, understanding that your background is, you're from a questioning background, and so once you came to that when you had that happen, that you were studying and you had the questions about the ribose and why is the ribose part of this most important molecule of life, can you take us along your path of discovery, or
Yes.
your work to today? Thank you so
Yeah.
much,
Yeah.
Doctor.
Definitely. So that's a very nice segue into how my thought process changed. So when you want to understand a structure, one of the things that my professor taught me was, you do not understand a particular structure looking at it a priori. For example, when we look at RNA, you can ask the question, why ribose and not any other sugar? Why phosphate and not any other linker? Or why the 2 purine bases and 2 pyrimidine bases? Why did nature choose a purine pyrimidine paradigm? Could it not have chosen 2 purine paradigms or 2 pyrimidine paradigms? Why this 6-5 membered with another 6-membered ring kind of combination? So when you look at this, when you want to try and find answers, just studying RNA does not give you the clear answer. But if you make a variation of it, so for example, you take the ribose and replace it with a hexose, for example, a glucose, what happens to the base pairing property of that Watson-Crick base pairing? Amazingly, when that's what Eschenmoser did, when he replaced the ribose with a hexose, like a glucose or a fructose, he did glucose and the various isomers of that, the base pairing really fell off. It became a very weak base pairing system. Okay, so then you can come back and compare it with RNA and say, aha, RNA seems to have this base pairing property which the other system doesn't. Therefore, maybe life, use this one. Not so fast, because when we went to the ribose, which is in a 5-membered ring in the RNA, which is called a furanose, and you can change it to a pyranose, which is a 6-membered ring. So it's the same ribose, but not in a 5-membered ring shape, but in a 6-membered ring shape. It's called the pyranose ribose. Then, when we did that chemistry with Professor Eschenmoser leading the way, what we found was that the pyranose form of RNA is actually much stronger than RNA itself. It's much more Watson-Crick selective. So the answer is not simply the base pairing strength that nature chose it by. It must have chosen it because it's optimal for what its function is. That means it's like the Alice in Wonderland paradigm, not too strong, like the pyranose form, not too weak like the glucose form, but just right in the ribofuranose form. So, that really began to tell me that you can't look at these molecules in isolation to understand why they are special. You have to see how they interact with the environment and how they interact with other molecules, because as they begin to function, it is the function that is responsible for the selection. Just because the molecule is present, it's not selected. It is selected because it functions in a way. A non-functional molecule or a non-functional part of the molecule is not selected. It's just like, when you get infected, your body does not know the answer immediately, and it produces millions and millions and of antibodies, and it's only one or two antibodies that really work, and the system immediately recognizes that and produces more copies of that work, and all the other things that didn't work, it does not produce anymore. So it is the functional aspect of these molecules which they express themselves only at the level of, when they are interacting, what we call a systems chemistry. In a system only the function is expressed, not in isolation. So that is the guiding principle, and that will be the kind of background I give for about 5 or 6 slides before I jump into the philosophy of how we approach chemically the understanding the origins of life.
Thank you so much. And that strategy is so, it's just a beautiful strategy to apply to anything really. That, when you said if we replace the ribose with something else, then what happens? Yeah, it's just such a maybe a different methodology than, or it's just great to have in the toolbox. It's a fantastic way to look at things and keep questioning, maybe honest and open-minded.
Yep.
So yeah, so thank you very much. And it's like, with art, you can, if you're creating a dance or music, you can vary time or space or
Mm-hmm.
energy. It's all kind of the same. So thank you for that. And at this point, the mic is yours to continue your talk, and you see your slides are there, and then we can have a Q&A following your discussion if you'd like, or if you'd rather
Sure.
have questioning drive your discussion. That's entirely up to you. Sometimes people put questions in the room chat for you, and we, the moderators, are happy to share those with you. So
Sure.
please, thank you
So,
and enjoy.
if you feel the questions, are appropriate, at that time, please read them out, and I'll try to answer them on the fly. If not, I will say I will answer them at the end of the Q&A, so, depending on the context. Okay, so I'm going to begin, and what I just want is like a housekeeping note. So what these slides are going to have a number of animations. So when you click on the next button and it goes to 2, you may actually be on the same slide, and a figure will appear, or image will appear, or a text will appear, or it will disappear and be replaced by another text. So I'm on slide 1, but when I click, it's going to be on slide 1, but you will see this image appear. So the image that shown is, what we understand of so-called Darwinian evolution, when you go from a single cell to multicellularity, and, to what we see as man today. But, and that is possible because we are able to go back and reconstruct that based on fossils. So if you click the next one, you will see the deconvolution appear. And then, that's because we can deconvolute by walking back with fossil records and try to, come up with this, a theory, and, try to, understand how multicellularity would come. However, if you click the next one and if you replace this, the images disappear, and now you are replacing it with, you know, prebiotic chemistry and biology and life, where you are replacing it with the molecules of life, then things get a little complicated because there are large gaps in our, if you click the next arrow, you'll see arrow with gaps in between, gaps in knowledge of how, the molecules went from being an amino acid to a protein, and then gave rise to what we call life today. And it's also because we have a lot of black boxes. So if you click next, black boxes would appear, and these black boxes not necessarily are, that I can't demonstrate. I can demonstrate. I can take mixtures of amino acids and make proteins, and then I can take proteins and put them somewhere and show life like property. But your question will always be, Ram, you have done this, but do you know this is exactly the way it happened 4 billion years ago on Earth? And we have no answer, definitive answer to that, because there are no chemical fossils, unfortunately. So we can't go back. So if you click the arrow, the going back arrow is also quite difficult. So coming either from the molecules of life or coming from life itself, either a top-down approach or a bottom-up approach, still leaves us with this question. If you click next one, it says, what or how can we know about the chemical origins of life? Remember, it's a question of can. I didn't say should, could or did, but I said can, and there is a reason for that. So when clicking next, you go to the second slide, and in the second slide, you have the three pillars of prebiotic chemistry. If you click once more, the three reactions will appear. One is called the foremost reaction. The other is the Urey-Miller famous spark discharge experiment, which converted gases like methane, ammonia, hydrogen in the presence of water to amino acids. And then the third one is not well known, but it is the polymerization of a simple molecule called hydrogen cyanide, which gives long black polymers, which then, if you hydrolyze, produces a lot of heterogeneous mixtures of molecules, one of which is adenine. There are a lot more, but there is adenine. So people look at this and they say, oh, these kind of simple reactions give rise to the building blocks of life. So life must be possible from this kind of chemistry. Not so fast. If you click once more, you will see the current state of the art of people's thinking. They will either rely on what is known as an RNA world, where people would hypothesize that RNA was the first self-replicating molecule that came about and then gave rise to everything else. Then there are people who say no, that's not right. There is a metabolism world where you have small molecules coming together. And then there are people who still argue for proteins appearing first, because amino acids are very elementary in prebiotic chemistry. And then there are people who say, wait, you need to put this all in an enclosure. So you need some sort of a vesicle, which the lipids provide. So it's a lipid world. And, they go on and on with this one. But the problem with all this is that they forget that we are working with really prebiotic mixture. So if you click next, you will see the term one among many compounds. So though you see ribose, though you see adenine, and though you see glycine there, or the amino acids, they are among a terrible mixture of compounds that are formed. So the question is, how does that, if you click once more, you will see, how does that prebiotic many become the emergent few? So how does the prebiotic, the biological few, how does it become that? And the problem with the strategy currently pursued by people who believe in the RNA world or the metabolism world is that they are trying to extrapolate biology all the way back to prebiotic chemistry 4 billion years ago, when there was no biology. So what we are trying to do is, we are taking a finished product and we are looking at the parts of the finished product and trying to find it in the wood that it came from. So you will not find it, unfortunately, because it is completely coarse, and many of the clues have been destroyed either by the earth being so violently changing in the early years, or by life itself. Life is life is so ubiquitous on earth, you will find it almost everywhere. And even if you find something, you have to prove that it's not from modern life, but something ancient that existed 4 billion years ago. And that is always a challenge. So clicking on that, you will see that, yeah. So for some strange reason, it's not moving. There are only 2 slides.
That was my question. Was about to ask you to check to make sure that it was. There are only
Catherine, you
two.
might have clicked the wrong slide. You might have put the wrong one.
Oh.
One which was the test slide that I sent you. Then I sent you the other one.
Okay, let me just copy again. Sorry about that.
No problem. I'll click done and then we'll click the next one. Yeah, those are the test slides. Oh, I didn't notice. I should have told
I'm
you.
so sorry that I couldn't. It was the whole thing of restarting the room the next day.
Okay.
So it should be the right
So
one.
this is just the first slide acknowledging all the funders and all my colleagues who worked on this. And so we'll skip to the third slide, slide number 3, if you want to go to. So just click slide 4. Yeah, we'll now go to slide 4. Okay. So if people are on slide 4, they should see the camel problem. Everybody see that?
Yeah, I can see it. Frank, everyone.
So
Yeah. Okay.
I will assume that everybody is on the slide number 4 where they see the camel problem. And this is an interesting question. So there is a problem. A father leaves 17 camels for his 3 sons, and when the father passes away, they open the will, and the math is a little difficult. He says half of the 17 should be given to the first son, 1/3 to the second, 1/9 to the third, and you know that you can't divide the camels in a proper way. Either you have a unhappy camel or a unhappy son. So then how do you solve the problem? So if you click next, you'll go to the next slide. And I'll tell you what the relation of that problem is to our thinking in approaching origins of life. So this is a quote from my professor, who's my mentor in this field. And he says the aim of such people like me working in this field is not to look at it as a historical problem, but look at it more like a challenge. Can you make life? Can your chemicals come together and make something called life-like processes? That is more important than trying to reconstruct the history, because as I told you before, we are struggling to reconstruct the history because we have very less clues to do that. So if you click once more, you will see what the approach that we take is called systems chemistry. So we try to mix molecules and see what sort of interactions arise under a given conditions, and it's shown in a way that the philosophy that we work with is the origins of life cannot be discovered because there are so many ifs and buts there. So can we reinvent it? Can we with our ingenuity or our learning and being humble through the learning process, come up with a paradigm, come up with a model that can? So the question that we ask is, so if you click once more, you will see colored balls appear with the prebiotic many. Can we take the mixture of chemicals under prebiotic conditions? Click once more. Can you come up with systems that emerge? And one more click will give you what we call a process called proto-life. So can we take this process in the lab and see what happens? So the idea is that if you click once more, it is an experimental approach that is based on emergent properties of naturally manifesting systems from a mixture. I don't engineer the system in any way once I mix it. I only define the starting conditions and I mix it and then see what goes on, just as an observer from outside. Okay. Then based on the observations, I can go back and reiterate, change the condition or change the ratios of the mixtures and see what happens. This is a non-teleological approach to biology, how properties emerge. And I'll explain what non-teleology means in the next slide. So click once more and you will see at the bottom some molecules appear. So let us say you are on a planet, exoplanet, where you do not know anything and you only know these type of molecules exist there. The question you should ask yourself is, based on these molecules, can you already predict what the biological molecule should be? Without knowing what the outcome is, how do I know I need to pick ribose, or how do I need to pick adenine, because I do not know what the end product is? Remember, you are on the earth 4.2 billion years ago and you still don't know what biology is because it has not yet started. How do you know to look for RNA? Because RNA is not there. Click once more. Do I make a selection at a more complex level when these molecules come together? One more. Or at a higher complex level, or at the oligomer level, what we call oligonucleotide level? One more click. Or do I make the selection at when the RNA emerges as a functional entity? So you can have selection at many different levels. One more click. So you can't select for RNA at the ribose level because there is still not an RNA, right? But you can select for something else based on the property of the ribose versus lixose or the other isomers of the sugar. One more click is, if you try to superimpose biology on prebiotic chemistry 4.2 billion years ago, you are doing so because you already know the outcome, and that's not the way to understand how life arose. If you already know the answer, then you will only try to engineer your systems to find that answer. Okay. So what I'm arguing is there is no teleology or purposeful destination when you start off in the very beginning. Therefore, when you click once more, there is a very famous saying by Francois Jacob who said, evolution is a tinkerer, not an engineer. And you click once more, you'll get, that's also true for chemical evolution. That means if you click twice, it's a systems chemistry approach. That means, can I mix these molecules, and would RNA be the only solution that I get at the end? If it is the only solution, then that's fine. But if there are other solutions that come up, I need to pay attention to that. And I'll give you one concrete example of how focusing on biology can mislead you. So click once more, you will see a molecule RNA, and you will see a molecule cytidine on the left hand side and uridine on the right hand side. These are the building blocks of RNA, and these are called pyrimidine nucleotides. And if you look and make a connection to prebiotic chemistry once more, click, you will see these, the uracil and cytosine, which are the nucleobases. They come naturally from these hydrogen cyanide polymers that I told you before. So people make a connection and they say, look, you know um, this can work so nicely. But the problem is, it's not simple, because if you look at how biology makes it, one more click, biology makes the uridine and cytidine starting with orotidine. It's a completely different molecule. And the nice thing about this, if you look in prebiotic chemistry, in biology, it takes it from orotic acid. So nature still today, does not make these uracil and cytidine. If you click once more, you'll see a green bar over the natural bases. But what it does is it makes only orotic acid starting from aspartic acid. So if you try only looking at RNA as the finished product and look at uridine and cytidine and look at the prebiotic chemistry, and you look only for those bases, but you completely ignore the orotic acid that biology is using. And that's my point. So click once more and you will see this, what I call the BBB syndrome, which is called the blinded by biology syndrome. We look at biology and we extrapolate it, blindly, what prebiotic chemistry must do in order for you to get life's building blocks. And I will show you an approach where if you ignore biology, you may be able to get at the same molecule, but through a completely different route. So if you click on the next slide, you will see the answer to the math problem. So there you see that there is the camel problem. And once you are not able to solve a problem, you generally go to a wise person. And they went to a wise person who said, oh, I'll solve it. I'll come with one more camel of mine. He adds it to the total. And then it becomes 18. And now he's able to do the math. So 18, so half of that, he gives it to the first son. Second, 1/3 gives it to the middle. And then 1/9 gives it to the second. And if you add the 9, 6, and 2, it's 17. So the man takes his camel back. Now, what does this mean for our chemistry on origins of life? My question is, by ignoring in prebiotic chemistry other molecules, remember the very slide, that first slide I showed you, the 3 reactions where there are mixtures, could those other molecules could have provided some solution and they have just gone away, just like this 18th camel? So if you click on the next slide, how does this 18th camel help? So you go to the slide number 8, where we come to the problem of peptide formation. So peptides are the building blocks for proteins, and they are nothing but 2 amino acid units joined together by what we call an amide bond. And people, remember the Stanley Miller, Urey Miller experiment, the famous spark discharge. People were excited finding amino acids. So they just said, let's take amino acids and try to make peptides out of them. Till today, there has not been a very good solution to that. Okay, because you have to do so many shenanigans to that and then come up with answers or justifications. But if you actually go and look at, if you click once more in the context of prebiotic chemistry and go back and look at the Urey Miller experiment, where you see this very nice spark discharge experiment that's still there at UCSD in San Diego, what it is it's not only those amino acids. You click once more, this reaction also produces molecules called hydroxy acids, where the NH2 of an amino acid is replaced by a hydroxy acid. And so now the question is, you go and look at the meteorites, where you have heard these wonderful news stories where meteorites bring the building blocks of life. But the building blocks of life are not the only molecules on meteorites. They bring all other kind of molecules. And if you look at the chemical inventory, there is also the same hydroxy acids that is produced in the Miller experiment. So when you look at prebiotic chemistry, it tells you that alpha amino acids are not alone. They are always produced with alpha hydroxy acids. And now the question is, why are we ignoring those alpha hydroxy acids? Because nature does not use them. So if you click once more, you will see that you'll see that the BBB bias we tend to ignore because it's not sensational. You imagine Stanley Miller would not have gotten the notoriety if he said, I produce alpha hydroxy acids, published in Science, and then New York Times wrote a big article about it, made him very famous. But he was very famous because he identified amino acids. But the hydroxy acids are still there. And now the question that we asked ourselves is, if you click once more, can the mixture give you the same peptides? Okay. Can you start with a mixture of alpha hydroxy acid and amino acids and produce the same peptides, which people have been struggling to do? So if you click once more, you will see the amino acid in blue and the hydroxy acid in red. And what we did was a very simple experiment. We mixed them in water, did nothing else, just dried them down at 80 degrees centigrade. And then once all the water was gone, we simply added water back and then repeated the same drying. And then we added water back and repeated the same drying. It's known as a wet dry cycle, which can be simulated on early Earth, by the sun beating down on a pond. The pond dries, then rain comes in, so it's a kind of a scenario. And what happened was, you see this red molecule that is drawn there. I can't unfortunately use any marker or presenter here, but the red molecule at the bottom is known as an ester bond, where you see this, two molecules joined together, and that ester bond is now active. And what happens is an amino acid attacks it and makes an amide bond naturally in that system without any, us doing anything. And then what happens is, you see that the blue has now come in and replaced another red molecule, and now you see there is an extra carboxylic acid at the end. It's called a deoxypeptide. And this, if you continue, will continue by itself. And before you know it, what happens is you get a long polymer which is enriched in peptide bonds. It's not a pure peptide, because it's a deoxypeptide, because it has hydroxy ester bonds in between. But what you have done, you have enhanced the peptide bond formation naturally, starting from a mixture. And what is so nice about that is, if you click once more at the left hand bottom, you will see this summary that peptides can naturally emerge from a plausible prebiotic mixture. And what is so pleasing about this demonstration, if you click once more, is what exactly your ribosome does when it's making peptides in your body. It makes an ester bond with an amino acid with what is known as a tRNA. It's called and then it brings the two tRNAs on a messenger RNA. And then you see the NH2 group now attacks that carbonyl group of that ester and makes a peptide bond, and the whole thing repeats again. So what we have done is, we have, discovered the same chemistry that nature is doing now, but in a very primordial sense, just with the, building blocks of just amino acids and hydroxy acids. And so now, if you click one more, you will go to the 9th slide. And if you keep clicking, you will see the appearance of these colored balls, and you keep clicking and you keep clicking. And then the question is that, can you do this evolution? And you click once more. The question you ask is that alpha hydroxy or 18th camel, which comes and goes? It's being recycled, right? So it is as if the 18th camel is that hydroxy acid which comes, solves your peptide problems, and then goes away. Exactly what, nature does with the ribosome. Click once more, you're on slide 10, and you'll see a railway line which is disconnected. And this disconnection shows the disconnect that we have in trying to bridge biology to prebiotic chemistry using only biological molecules. On the other hand, you click once more, that figure will disappear. Click once more, you'll get, so now if you introduce the hydroxy acid, which is in the green, and you click once more, you get a different railway line. One that is not completely disconnected, but one is not completely connected either. It is it's like you have to change stations somewhere in between and change, from one track to another. And this is not unrealistic. If you look in the history of the world, about 2 to 2.5 billion years ago, there was an abrupt rise in oxygen. And that caused a lot of issues for the chemistries that could not survive oxygen, and those died off. Those that could survive had to adapt or invent new things. So if, for example, if you look at photosynthetic oxygenic photosynthesis, that will go only back to about to 2.5 billion years back, not before, because previously there was this anaerobic or anoxygenic chemistry that was occurring. So one has to be very careful of how you extrapolate biology all the way to prebiotic chemistry. Okay, so that gives you an example of how you can start with different prebiotic chemistry and switch to different biology, but some semblance of connection with the same amino acids and the same mechanism that is common between what I showed with the ribosome, ester mediated peptide, and the ester mediated with the amino acids and hydroxy acids. So clicking on it, you go to slide 11. In slide 11, the metabolism starts. So I will stop here and ask if there are any questions before I delve into the metabolism aspect of this one.
Yeah, please go ahead and ask.
Okay.
Oh, Frank,
Yeah.
go ahead.
Yeah.
Uh,
So Frank,
yeah,
any questions?
So,
Yeah.
so it's definitely a treat with this new set of slides. Much better than the previous, just only two. So it caused me a lot of panic. I was thinking, should I go do the original paper? So
No,
that's
no,
why
no,
I
no,
missed
no,
the
no.
question. But
Sorry about that. I apologize. Yeah.
that's it works great. It's just like this is very information dense. By the way, the way this is the first time I saw the way it's very ingenious that you use the fully utilize the PPT, the animation feature that go step by step. But just quickly on the big message you are trying to deliver here, let me see if I get it right. So for the prebiotic, that's very intriguing to see. There's something, the 18th chemo that comes and goes, i.e. the hydroxy acid. So when you say emphasize being the word can, that means they so I was distracted a little. What happens is it when you say can, that means that if the condition back then was a little bit different, then we have the different track
You are
track.
wonderful question, really, Frank. Wonderful question, and you are jumping way ahead of me. I'm going to address that in the very last slide. Can you wait till the last
Well,
slide?
I just want to make sure that I got the.
You are
That
absolutely
you're
right. You
implying
are
that
absolutely right.
I got I'm on track, right? But still, the when you say the wet and dry cycle, that's quite because one of our hosts is a very is expert on this topic, but she wasn't
Yeah,
here today. So there's
yeah,
the clay, there's competing hypothesis, right? Thermal
correct.
vents versus
Yes,
the wet dry, like lakeshore or seashore.
correct. Yeah,
There's a clay, there's a
yep,
matrix to
yep,
it. So
yep,
do you have to take all these? So wet dry meaning that it's at least what I read is the facilitates the membranes formation. So there's a um, their demarcation of inside and out. So anyway, that's probably just one point, looking at the whole elephant through a speckle, a
yeah.
microscope. Yeah.
You're absolutely right, Frank. And thanks for bringing that up. As I pointed out before, there are many theories out there and many ways to approach this problem. And I would be remiss if I claim that mine is the only way to do this. Not at all. So what I'm just showing here is a proof of principle experiment that if you start with mixtures, the answers emerge naturally rather than trying to force the answer only through looking through the lens of biology. To this you can add metals, you can add clays. We have done that. So one of my colleagues is looking at what all could be added to this mixture and how they would influence the outcome. Would there be some sort of an absorption on the clay that would lead to selective absorption of the peptides that would form? Can we recycle and make longer peptides? All of those are valid questions. We have not looked into it. I'm very much interested in showing the proof of principle, and and then taking it further to a higher level, rather than just getting caught in that conditions, because that could be a jungle where you get caught, and you can find yourself in a local minima that you are not able to come out of it at all. And many people are stuck in that minima like that. They focus only on their pet theory of clays or thermal vents or wet dry cycling, and then they are not able to come out of that, because then they throw everything according to that wet dry cycle or the thermal vent theory, and everything has to happen there. They just don't open up a little bit. And there are problems with each and every approach. There are drawbacks with each and every approach. But what we try to show here is that you starting with mixtures, you are better off. And this is not just for peptide. We have also shown it in oligonucleotides. And I'm not going to talk about that because the focus is on metabolism. But I use this as a principle, show as a proof of principle, the philosophy underlying our approach in trying to understand how mixtures can give rise to more clean materials or more homogeneous materials. That way, because life relies on these homogeneous materials. So if you take a peptide, it's not homogeneous in terms of composition, but it's homogeneous in terms of its peptide composition, that is the peptide bonds. The same way, if you take oligo, it's not homogeneous with the AUGC, but it's homogeneous with respect to the phospholipase backbone. So those are the kind of things that we are trying to develop, so that we are then trying to see whether that emergence leads to more interactions or different type of interactions that give rise to lifelike properties. But your point is well taken. There are many theories out there, but we do not try to get caught in one of them alone. That's my point. Okay.
Got it. And it's an excellent answer, and very much looking for. I mean, this is definitely. I somehow I think this. I subscribe to your philosophy as well. It will be a productive one. Get a result and cutting the garden knots. Yeah. So great. Looking forward to the more to come.
Yeah. Thanks, Frank. So I'll go on to slide number 11, which now, now we are talking about metabolism, and people are trying to do the same thing. They look at metabolic pathways in life today, and then they look at starting molecules on Earth, and then they say, let us try to make a direct connection. And there have been a lot of papers. And one of the most important cycle that has been studied, if you click once more, is called the reverse citric acid cycle, shown on the top right-hand corner. It's a very complex cycle, which is run by many enzymes. And I will tell you that, it's almost impossible for a chemist like me to envision running this all reactions in a single pot without the help of enzymes, because there are so many competing reactions, as I will shortly allude to in the next slide. So you click once more. So people have taken that, and you will see a simplistic representation with black dots, two type of circles. So people have really looked at this and said, let us simplify this and see if we can start with carbon dioxide. So the argument is based on, if you click once more, the yellow carbon dioxide as a building block, parsimony argument will appear. So people want a continuity right from the beginning to the end. They don't want a break, as I alluded to in the previous slide. So they want to say, okay, if I do everything with carbon dioxide from the beginning, then it becomes easier for me to transition to what life is doing today. I don't want to use cyanide. I don't want to use anything that life is not using today.
Ram, quickly, I just do housekeeping. For the new audience, we are on the number slides number 11, and probably at the last click. It's each slide has multiple clicks, so if you want to follow,
that's right.
Thank you.
Yeah. Thanks again. Just to reiterate, each slide has a number of figures. So when you click, you may not automatically go to the next slide, but images will appear or disappear and appear based on the sequence that I have put. The reason I have done this is because if I just show the slide fully, it will be too cluttered and people will get confused, especially when I'm going to go to the later slides where there's a lot of organic chemistry structures, and I apologize in advance for that. But as an organic chemist, I can't escape. And as a life that's based on organic chemistry, we can't escape either. So click on that again, and now you go to slide number 12. And here you see the reductive citric acid cycle in its full glory. And this is what people have focused on. And the reason they focus on this is because it takes carbon dioxide and converts it into organic molecules. Okay, so it's an anabolic cycle, not a metabolic, catabolic one, but an anabolic. It builds from carbon dioxide. Okay, so there are many steps where carbon dioxide is taken in, especially where if you look, let me see, unfortunately, I can't use this pointer or anything, but going from oxaloacetate to α-ketoacids, if you can see that, it goes by, from succinate. At the very bottom left hand side, you will see succinate. Succinate goes to succinyl CoA, and then it goes to α-ketoacids. So that, is building up, or incorporating a carbon dioxide in there. So what many people have tried to reproduce this, and they have failed, mainly because it's very tough in order to run this whole cycle in a single pot without the help of enzymes. And this, if you click once more, was pointed out by Leslie Orgel in a very famous paper where he argued point by point, saying that how many, there are so many competing reactions. There are so many, molecules that look similar. Without an enzyme, you cannot discriminate them. It becomes very difficult. So what we decided to do was, you click once more, is that, you'll see that there are side reactions. So you click once more, you will see what we decided to do was, instead of focusing on the molecules, we focused on the reaction types. As an organic chemist, I can break this reductive citric acid cycle into very simple reactions, such as hydration, dehydration, decarboxylation, carboxylation, what is known as a carbon bond forming reaction called the aldol reaction, or its reverse breakage called the retroaldol reaction. So these are the simple things. And then we said, are there simple molecules that we can try to reproduce these chemical reactions and see what happens? Not the citric acid cycle, not the reductive citric acid cycle, but what chemical reactions can happen. So we use these two molecules called the pyruvate and glyoxylate. These are two molecules that exist in biology today. And then we also used a molecule called malonate that also exists in biology today. And these are systems that are quite reactive because they have these kind of carbonyl groups next to each other. So if you click once more, what we did in collaboration with Professor Greg Springsteen at University of Furman is that we let these molecules react, and you click once more, you will see some cyclic reactions appear. I will not go through them in detail, but just suffice to say that we just mix them in the pot and add chemicals, and we can begin to see these kind of reactions and monitor them by analytical techniques, which gives us the confidence. For example, the cycle on the right hand side, which is called the malonate cycle, we can run them many times, about 3 times at least, by using C13 label isotope labeling to show incorporation of more and more labeled compounds into the products. We also did what was known as so if you just mix the pyruvate and glyoxylate just by itself in buffer, the reaction happens and we produce a series of chemicals which are nothing but the alpha keto. It's an analog of the reductive citric acid cycle. And what is important is that green circle that is there with glyoxylate. Actually, that's a reduction of the double bond of that molecule to produce α-ketoacids, which is an important part of the TCA cycle. So here we have not added any metals. Nature uses metals to do this reduction, or uses NADH and cofactors to do this. We have simply produced it by using a simple organic molecule, which is also the starting material. And I won't go into the mechanistic details, but it is through what is known as a cross Cannizzaro reaction, or it's a retroclaisen reaction. There are two mechanisms that happen. I will not go into it, but this is what we found. That means by simply mixing the two chemicals, we can produce a series of reactions that almost simulate a reductive citric acid cycle. And we didn't want, that was not what we were looking for. We were just exploring. So this is something that naturally emerges when you mix these two. Okay, you don't need any complicated enzymes, you don't need any complicated metals to do this reaction. You just need these two chemicals in a buffer around pH 7, pH 8 or 8.5, and you can heat it to room 45-50 degrees. You leave it at room temperature, the reaction goes very slowly. This, if you want to run it in 24 hours, you just heat it to 50 degrees. So clicking on that, you go to, so what does this mean, in the context of what we are trying to prove here, or show here. So you click once more, you go to slide 14. So what we wondered was, what happens if we introduce cyanide into the system? And the reason we had thought about that was because we discovered this reaction in 2017, where we showed if you mix cyanide and glyoxylate, it makes an adduct immediately called the cyanohydrin, which is shown the second molecule. And it doesn't stop there. It immediately reacts with another molecule to make a more complex molecule. And that's a cyanohydrin, that's the third molecule from the left-hand side. And for us, it was a shock to discover that cyanide, usually cyanides are very difficult to hydrolyze, but the cyanide hydrolyzed spontaneously at room temperature, slowly though, to give the corresponding carboxylic acid, which we could just decarboxylate by heating and make tartaric acid. So you could take two molecules of glyoxylate in the presence of cyanide and make tartaric acid in the single pot. It's a very clean reaction. Um, what was surprising for us was we had done a reduction. We have taken a carbonyl group, which is circled on the red on the glyoxylate, and converted it to an alcohol group, which is a reduction, using simply cyanide. So what it gave us was that, it is going through this kind of a mechanism for people who are mechanistically inclined. It's a hydrolysis that takes place via this carboxyl accelerated, and that becomes important. If you count the number of atoms 1, 2, 3, 4, 5, that 1,5 relationship is very special. Okay, so if you click once more, the green sentence will appear saying important 1,5 relationship is going to be critical for selectivity. Those molecules that do not have this 1,5 relationship will not be hydrolyzed, and you will begin to see the value of this when you apply this to the citric acid cycle. So what we did was, you click once more, we went to the citric acid cycle and said, what are the molecules that our cyanide can interact with? And so if you click once more, you will get a list in blue of all the citric acid cycle molecules that are involved in the citric acid cycle. And if you look very closely, all of them have this carbonyl group or a carbon-carbon double bond group. For example, the pyruvate, oxaloacetate, which are the first two molecules, alpha-ketoglutarate, which is in the middle, fumarate, which has a double bond, which is the fourth molecule from the left-hand side, the aconitate, which is the second molecule from the right-hand side. Those all have what we call unsaturation centers that can react with cyanide. And now let us see what happens when you let cyanide react with them. So click once more. I just now have highlighted the molecules that can interact with the cyanide. Others cannot because they don't have the unsaturation. So now we throw in cyanide with oxaloacetate, which is the second molecule. Click once more, you'll see an arrow. And what that does in the presence of cyanide, click once more, is it makes the cyanohydrin as we saw in before. But now, because you have the 1,5 relationship, it'll hydrolyze it and gives the corresponding dicarboxylic acid of that, which you decarboxylate. It gives you malate, which happens to be the next product in the reductive citric acid cycle. So it happens automatically. I don't have to use any special enzyme. Now, if I do that with pyruvate, you click once more, which is the molecule at the left-hand side, it just makes the adduct, but since it does not have the 1,5 relationship, nothing happens. It just keeps going back and forth. Now you click once more, the same with the alpha-ketoglutarate, which is the molecule, which is the fourth molecule from the right-hand side. Nothing happens to it because it just adds, and then because it doesn't have the 1,5 relationship, it just simply goes back and forth. Now let's come to the fumarate. So if you go to the fumarate, what you see is, if you click twice, you will see that the fumarate, again, it can add, but now it has the 1,5 relationship. It can make hydrolyze, and then you decarboxylate and gives you, the next molecule, which is succinate. It was simply amazing for us that a simple molecule like cyanide could differentiate between the different constituents of citric acid cycle and only do the reduction exactly as what the enzymes do today. So a simple molecule cyanide can make the selection for you among the mixture of molecules. In fact, we have shown that if you take a mixture of these molecules, that's exactly what happens. Only the molecules that can be reduced or hydrolyzed and reduced get that. Others just make the cyanohydrin adduct and then stop there. So what about isocitrate, which is the aconitate, which is the second molecule from the right-hand side? That also can react. And it makes this, if you double click and you go to the very end, it goes and it makes what is known as a tricarboxylic acid, which if you want, you can oxidize to citrate, but that's not of interest for us. But it also does the interaction. That's what we are trying to say. So if you click once more, What it is you will see cyanide is a primordial reductant. It can act as a primordial reductant, as an abiotic reductant. And it addresses, if you click once more, it addresses the selectivity issue that Orgel brought up before. And it is, it's a very, pleasing way for us, at least intellectually, to discover that a simple molecule can do the job of a complicated enzyme in this kind of a mixture, in a what could be a simplistic way. And therefore we can use this as a system to probe further what can be done in a non-enzymatic fashion, which is what we think happened, or could have happened or can happen in an abiotic sense. So click once more, you go to the next slide. The problem, now is with one of the molecules of the TCA cycle. It's called oxaloacetate. That oxaloacetate is a very important molecule. That is the one that goes further to give you malate, and then dehydrates to give fumarate. But the problem is, if you take this oxaloacetate, which is commercially available, and put it in water, it will decompose very rapidly to make pyruvate. That carbon dioxide is lost, and it's very difficult to put that carbon dioxide back. People have tried to take pyruvate and put carbon dioxide and make oxaloacetate, and nobody has succeeded so far because the reverse reaction is so fast. The other one is thermodynamically uphill reaction. Click once more. So the question we asked is, can we bypass this oxalate completely? So if you look at the sequence of reactions, now again, this is going to the philosophy that we are not going to be dictated by what we see in biology, but can we bypass the whole thing, and come up with simpler systems? And that's where I'm taking you. So you can take glyoxylate and mix it with this molecule called malonate. That is an acetate equivalent. Nature uses acetyl-CoA. Okay, so we are using malonate as the prebiotic or the abiotic equivalent of acetyl-CoA. And then you click once more, this acetyl-CoA, this malonate combines with glyoxylate to give this aldol reaction, a carbon-carbon bond forming reaction. And then you click once more, that can give rise to two different products. If it loses carbon dioxide, it gives malate. And if it loses water, it gives us what is known as the carboxyfumarate, which is the molecule at the bottom, which is the black. Now you can increase, react that with, you can also dehydrate that malate to give you fumarate, which is part of the TCA cycle. And if you add cyanide to that fumarate, which we have already seen, that gives you carboxysuccinate. Okay? Now we can also do the same thing to that other molecule, carboxyfumarate, which is at the bottom, and go to the same molecule. And this molecule, carboxysuccinate, turned out to be extremely crucial for the next discovery that we made. This molecule can now react, it can decarboxylate and go to succinate, which is part of the blue citric acid cycle, or it can combine with another molecule of glyoxylate, click once more, and make what is known as a carboxy isocitrate. And you see that red carboxy group, you can decarboxylate, you click once more, and it gives you isocitrate, which is what the RTCA cycle makes. You can't do the same reaction with succinate and glyoxylate because the succinate blue, which is right above the isocitrate, is inactive, it's not reactive. But the carboxysuccinate, where it comes from the loss of the carbon dioxide, called the carboxysuccinate, is reactive enough to do this. And then if you do a retroaldol, a breakage of that isocitrate, you can go back to glyoxylate and succinate, and the glyoxylate can be recycled into the reaction. Unfortunately, we have not shown that last step because the conditions under which we do that breakage, if you click once more, is that the glyoxylate immediately does a Cannizzaro reaction and makes glycolate and oxalate. And recently there has been another paper published by a different group where they take that glycolate, which is the bottom molecule, and convert it back to glyoxylate. So this type of a cycle of reactions can emerge naturally in a single pot. And we have shown that we can actually do this. And if you look very closely, click once more, this cycle has no oxaloacetate or alpha-ketoglutarate as the RTCA cycle. So we have developed a much simpler cycle which completely bypasses the two most problematic molecules to be handled in prebiotic chemistry: oxaloacetate from a stability viewpoint, alpha-ketoglutarate from a reactivity viewpoint. So we have bypassed it and we now have a cycle. Okay, you can say, great, you have a cycle. What does this mean? What is its significance? So we can do this in one pot. That means we can just take glyoxylate, malonate, and mix it with cyanide, and we see all these species emerging by an NMR, analytical spectroscopy, so we can see that. So that means we can run this reaction, and all we need to do is find conditions for completing the cycle so that we can go around and around. But let us say we do that. What is the significance of that in the context of origins of life? So click once more, and you will see the cycle that I showed on the previous slide on the left-hand side. So this is the protometabolic pathway that is mediated by cyanide. Okay, this is what I showed you before. And now let us juxtapose this. Click once more, and you will see the, click once more. On the right-hand side, you will see the citric acid cycle mediated by enzymes, by complex enzymes in biology. So the right-hand side is the one that exists in biology. The left-hand side is the one that we have demonstrated in a lab using cyanide. So now let us make the comparison between the two. Before that, remember that the biological cycle, you click once more, has what is known as a glyoxylate shunt, where the isocitrate breaks down, and then the glyoxylate mixes with acetyl-CoA and gives you malate, and the malate is oxidized to oxaloacetate, and that is an oxidative glyoxylate shunt. And this is known in biology as well. So now let us compare these two systems and go systematically, molecule by molecule. So if you look at the isocitrate, if you click once more, there is a box that will appear, which will show the box of the molecule on the left-hand side, correspondingly on the biological side on the right-hand side. So you see the isocitrate, which is common to both of these cycles. Okay, click once more, you will see the glyoxylate and succinate, which is again common to both cycles. Now let's click once more, and now you'll see malate, which is common to both of them. And once more, you will see the fumarate. So you see there are a lot of overlaps from the simple cycle we have developed and the simple, the complex cycle that biology has. The odd man out for us is this carboxysuccinate, which is now in a cycle, which is in a circle, and we do not have the same equivalent on the biological side. Okay, now we are going to click in a series so that we begin to see the arrows appear of how this conversion happens between these molecules. So click once more, you will see the glyoxylate going to malate, okay, on the left-hand side. You click once more, the same arrow will appear on the biological side. Okay, you see that, do you see that, that's not coincidence. But now the change happens. You click once more, on the left-hand side cycle, you will see the malate going to fumarate. But now if you click on the biological side, you see you're turning the other way around. You're not going the oxidative cycle. You're going this way around. You are coming to the fumarate. Click once more, the fumarate gives succinate. Click once more, in the biology, you have the same one, right? And now the fumarate, the succinate, fumarate goes to combining with the, going to isocitrate. You'll see that as well. So in our side, we are using the carboxysuccinate to do that. And then there is the retroaldol of the isocitrate breaking down to glyoxylate, which is exactly what biology has. So by this juxtaposition, what we have discovered by looking at the biological cycle, by comparing it to our simple cycle, is we think we have discovered another cycle that has not been recognized so far, what we call the green arrow on the right-hand side, the green cycle completed by the green arrows on the right-hand side, which we call the reductive glyoxylate pathway. Click once more, you will see the term reductive glyoxylate pathway within that green cycle. So it is a third cycle that is hiding in between, which we think might have been much more ancient than the original reductive citric acid cycle itself. So click once more, and we ask the question, tantalizing questions. Could there have been a reductive glyoxylate pathway? Can there have been a reductive glyoxylate pathway which gave rise to this reductive citric acid cycle and then disappeared? Just like that crossover that you saw with the alpha hydroxy acids and deoxypeptides going to the amino acid, is this the 18th camel that could have given rise to the reductive citric acid cycle? Click once more, you go to that slide 17, where you see that depicted pictorially, that now with this component of a reductive citric acid cycle, you ask this question, could that have been the forerunner for this more, ancient reductive citric acid cycle, because remember, without the enzymes, you cannot handle oxaloacetate very, efficiently. It will decarboxylate to give you pyruvate. Okay. I will show you an example where we can break that rule in the next part of the topic. So if you click once more, you will come to this question, so are these protometabolic pathways useful for chemical evolution? Yes, you have given us something to think about, but can your cycle that you showed, this reductive glyoxylate pathway, can it move us the same way that the reductive citric acid cycle moves us, say, towards production of amino acids? That is going to be answered in the next part of the talk. I will stop here and let you ask any questions or any logistical issues before I proceed to the last part of the talk.
Yeah, thank you so much. Does anyone have questions? I know there are a few questions in the chat, but I think they're more for later on and not, to understand like the mechanisms themselves.
Okay. So I'll continue then. So click on the next one and we go to slide number 18. So now we start towards the amino acid synthesis. And if you go and search for amino acid synthesis in prebiotic chemistry, the only reaction that you will see mostly, or 99%, is the Strecker reaction. That's a very famous reaction discovered by Strecker way back in the late 1800s. And what it is a combination of aldehyde in the presence of cyanide and ammonia. If you click, that is the reaction A that will appear, and makes a amino nitrile, which then gets hydrolyzed to give you the amino acid. Till today, if you go and look at that condition for hydrolysis, it's like with almost boiling acid, boiling HCl. There are no good ways of doing this. People have begun to worry about it, and one of my colleagues has shown that you can use sugars to do this, but still it's not very clear how that happens. What if you click once more, biology, so you compare it to biology at the bottom. Biology uses alpha ketoacids and does what is known as a transamination. It takes an amino acid, transfer that nitrogen group, and produces another keto acid, and in the process. So it just transfers one amino group from an amino acid to the keto acid and takes that keto group from that to itself. So it's what is known as a transamination. So you can see when you compare reaction A to reaction B, they are completely different, right? So my question was, if biology has to develop this, when did biology develop this transamination? Or can we use keto acids itself in prebiotic chemistry? Is that possible? So you click once more, and we proposed, this is the proposed reaction that we had using a molecule called diamido phosphate, or DAP for short, which is a diamido equivalent of phosphate molecules. That is the molecule which has the two blue nitrogens on the left hand side reaction top. Okay, so what we had proposed was this kind of reaction. I will not go through the details because it did happen this way. We still got amino acids when we mix them, but actually it went through a different pathway. So you click once more, it'll vanish and you will get the new, what we call the observed Bucherer pathway, not the proposed. So what happens here is you add the cyanide, it makes a cyanohydrin. You add the ammonia, you make the amino nitrile of a keto acid, but then it closes under a special condition to give this molecule known as hydantoin, which then hydrolyzes to give you the amino acid. And I will take you through these steps in the next slides as to how this happened. This reaction is very wide with respect to the different nitrogen sources. It doesn't have to be ammonia. It can come from various different sources. All it needs is an amine to start off this whole process, which converts itself to ammonia. That's all. So click once more, we'll go to slide number 19, and we'll go through the details of how this reaction happens, because it's very important to understand, how this differs from the classical Strecker reaction that people have proposed. So what we have is the pyruvate. So the pyruvate, when you throw in this, ammonia source and cyanide, gives you a very good amount of the hydantoin of corresponding what we call alanine, which will go to alanine. So if you heat that molecule, that gives you what is known as an N-carboylated alanine, which loses that group and gives you alanine as the end product. You do this, and this is just the NMR just to show you that it is real. It's a very clean reaction, not a messy one at all. This is just a proton NMR showing how clean that reaction is. And the way it happens, you click once more on the right-hand side, you will see a sequence of reaction appears. So what it is the cyanide adds to that carbonyl group, start from pyruvate 1 and go upwards in the cycle. Cyanide adds, it makes molecule 2. Then the ammonia reacts with that to make the corresponding aminonitrile 4. If there is no carbon dioxide in that reaction, the reaction just stops there. But the moment you put a touch of carbon dioxide, either through a carbon dioxide balloon or bicarbonate, immediately that molecule number 4 is carboxylated, and then it immediately undergoes a series of reactions which takes you to the 5-methyl hydantoin number 6 at the bottom of the cycle. And that sits there till you hydrolyze it to release alanine. So this carbon dioxide is very important for this reaction. Without the carbon dioxide, this reaction does not happen. And on early Earth, there was plenty of carbon dioxide. So we don't have to worry about the availability of carbon dioxide for this reaction. Clicking once more, you go to slide number 20, and you can do this from glycine. There's a glyoxylate. You see glyoxylate going to glycine, and there is the NMR proving how clean that reaction is. But what, you can see that. And then what we also saw, to our surprise, if you click once more, is you started seeing a molecule called orotate, which is molecule number, I think it's 18. I'm not able to see it very clearly. Yeah, it's molecule number 18 on the right-hand side. And that molecule is very important because that is the same molecule, if you remember, is produced by biology, which then attaches itself to the ribose and makes uridine and cytidine. So we are, reproducing, and this is exactly how the Krebs cycle works. The Krebs cycle produces these, aspartate, and then converts aspartate to orotate, and then the orotate goes on. So we were really excited to see this naturally appearing in this system, this orotic acid. So that means we are reproducing some of the molecules and reactions of biology without even asking for it. This seems to be a natural end product of this. So, sorry, going back to that, okay, let's see, done. Let me go back. Oops. Let's see. Something happened. I have to go back to the correct slide. I do not know whether, let me go to slide number 15. No, slide number 20. Yeah, so we were on slide number 20. And so if you click once more, you will see glycine from glyoxylate, but with an added flavor. So it goes through that intermediate that I'm showing there, which is number 23, which rearranges to form the orotate through a completely different mechanism. This is not known in biology, by the way. So we were very excited. So we just, if you click once more, the question comes, what do these protometabolic pathways provide for chemical evolution? What it provides for is protometabolic pathways leading naturally via the amino acids to the nucleobases, just as you see in biology. Okay, so this is a mere coincidence. I'm not claiming that this is how exactly it happened, and I'm rediscovering what happened on early Earth. Not at all. It is just a mere coincidence, and it is very stimulating to see that coincidence with biology. That's all. So don't try to read too much into it. I don't. Clicking once more, you go to slide number 21. So what about oxaloacetate? Remember how I told you oxaloacetate is a problematic molecule? What happens if you try this reaction on oxaloacetate? Many people have tried to do the transamination on oxaloacetate. And if they do that, if you click once more, all they see is alanine and not aspartate, because as I told you before, the oxaloacetate decarboxylates to give pyruvate, and pyruvate will transaminate to give you alanine. But what about our case? Our case is actually different. If you do the reaction, what happens is, because cyanide is a very good nucleophile, it immediately traps that oxaloacetate before it decomposes and makes a stable cyanohydrin adduct. If you click, that will appear. Click once more, that cyanohydrin adduct in the presence of ammonia will make the corresponding aminonitrile adduct without decomposing. Click once more, that makes this important molecule. This is the hydantoin of aspartate. This is an important molecule, and I'll tell you why later. And this can open up and dehydrolyze to give you aspartate. This is the first example known under plausible periodic conditions of conversion of oxaloacetate to aspartate. Nobody else has been able to show this in a good manner. The yields are very, very nice. And if you click once more, you will see the crude NMR, the crude of this reaction, and it's a very clean NMR. It's a very high yielding reaction. So the presence of cyanide and ammonia can prevent the oxaloacetate decomposition if ever there was an oxaloacetate. I'm not claiming the oxaloacetate was available because it would be very difficult to preserve it. But if it was available, then this reaction would convert it to this, hydantoin and keep it safe. So I will not, the other NMRs are just to show by proof that we did, make it. You click once more, there is a summary of sentences that appear, and you can read through it. But the gist of it is that it is, it's a simple reaction, and it is various sources of ammonia. It happens under a wide variety of conditions, and all of these conditions could be connected to prebiotic chemistry. But what is more important is that it seems to be coincidental with the type of reactions that happen in biology. Okay, so click once more. Again, the same questions, but what do these protometabolic pathways provide for chemical evolution? We are in slide number 22. Click once more. There is a summary of the reactions that appear that I've shown you so far. And what becomes very important are these molecules, if you click once more, where they are boxed in a blue box. And those molecules are very important because if you look at them very closely, there is only one molecule that can go on to react further to make cyclic structures. Click once more, and an arrow will appear telling you which one. And that is important because if you click once more, you will see what happens that can cyclize to give you a molecule called dihydroorotate. And incidentally, this is exactly what happens in biology. It makes aspartate, gets N-carboylated, makes this N-carboyl aspartate, which is molecule number 15, and then it cyclizes to give you dihydroorotate, which then, if two hydrogens are removed to make orotate. And if you click once more, you will get the NMR spectra of this crude reaction, which clearly shows the formation of this molecule. Okay, and click once more, you will see that is what biology does. Biology takes the dihydroorotate and oxidizes to make orotate. That's how biology makes it. Okay, we have shown, if you click once more, alternative ways of making the orotate as well, starting from glycine and the simple hydantoin molecule number 28. So there are various ways with which you can begin to see how this matches with what biology is doing. I'm saying that because that is what people are interested in, but I'm not making the claim that this is what transitioned to biology. Okay, so let us separate that. What I'm trying to say is I can discover chemistries naturally emerging without wanting to imitate biology. I didn't want to imitate biology. I was just exploring reactions, and these things happen naturally. They pop out naturally, which happens to have coincidence with what biology is doing today. Click once more. But this gives you a purely chemical reason why pyrimidines are the only molecules that come out of the reverse citric acid cycle. Reverse citric acid cycle does not make the purines. Remember, the only nucleobase that is made by biology is this orotate molecule number 18. And you begin to understand why, based on a purely chemical reason, not necessarily based on a biological reason. Click once more. This provides alternative pathways, and so now you can start from the keto acids themselves, not from aldehydes, and continue with this. Click once more. You go to slide number 23, and now you say, but Ram, you use oxaloacetate, but you told us oxaloacetate was not available. Well, I can come up with a different way not to use oxaloacetate and still make the pyrimidines. Okay, so because, as I said, click once more and you'll see formation of oxaloacetate and its preservation under prebiotic conditions is not yet resolved. So remember this, you click once more, you will see the reductive glyoxylate cycle that happened. We use that as the guide now. And what we do is we take that glyoxylate and the malonate and we do this reaction. So if you click once more, you will see appearing on the right hand side the reaction between glyoxylate and malonate in the presence of molecules like urea or other amine sources. You get this molecule, the amine adds, the urea adds, and then it cyclizes and gives us the same dihydroorotate in good yields. So you can make these molecules from a mixture of compounds, and they are naturally forming. I didn't do this with the purpose of making it. These are all naturally occurring reactions when you mix them under a given set of conditions. Okay, so there is the NMR. You click once more, you will see the NMR showing the formation of these molecules and it's a decently clean reaction, not much side reactions happening. Click once more. So now we are going to go through and try to connect all the things that we have learned so far. So can there be natural emergence of interconnected protometabolic pathways in this type of primordial or simple abiotic chemistry? Click once. So that will tell you, the sentence will tell you the alpha ketoacid reactions. So you click once more, you will see that the reaction of cyanide alone with the ketoacids, the reaction of alpha. I'm surprised why the one. So this I'm sorry, the reaction should, the color should have matched. So you click many times and you will begin to see the blue cycle of reactions that gives rise to oxaloacetate. And then you click once more, you will see how the cycle begins to grow. Click once more, you will see how this and click once more, you will see this, and you will begin to see that you begin to produce these kind of complex reaction cycles that begin to happen. And you click once more, you will see the connection of how they can feedback with engineer. And the nice thing is, if you go to the very last part of that, it says under similar sets of conditions, water or phosphate or carbonate buffer, pH 6 through 9, room temperature to 80 degrees, you can either do wet dry cycles or you can leave it in solution. These kind of reactions work in solution. Some work better with wet dry, but you can also do it under solution. It takes a longer time. All these things point out to the need of catalysts. So what we are missing in this whole set of things is catalysts. And the idea is, do the amino acids produced in these reactions, can they begin to make peptides and then feed back as catalyst, catalyzing some of these reactions? That's where we are going. Can small peptides be coming from the type of amino acids that are produced in this reaction mixture? So click once more, we come to the last slide. And this again goes back in last slide, in the sense, there are some philosophical statements there afterwards, but this is the last, slide with respect to the chemistry of it. This is a central problem in origins of life. Click once more. Is that what is demonstrated by prebiotic chemistry generally is not what biology is based on. For example, formaldehyde or cyanide, biology does not use that. And when you look at what biology is using, you can't try to reproduce that in prebiotic chemistry, because they were all done by complex enzymes, all the transformations. So the question then becomes is how to bridge the gap. So you click once more, you get that question. And the answer for me would be that the structures and chemistry used by extant biology must have evolved over time with the changing environment and the changing properties of those molecules. You can't escape that. Your molecules in a given environment express a property, and that's the property that you need to work with, because that is what will determine what the function is. So click once more, you will see the human evolution picture. And what I'm trying to say is, if this sort of an evolution is possible, you click once more, you will see the colored balls appearing all the way. And what I just say is that you also can try to do this in a chemical evolution approach, what I call chemical evolution. You click once more, you go to slide number 26, which now asks the question, showing this railway track, a disconnected railway track, saying that how similar should the chemistry of life be to prebiotic chemistry? Should it be the same? Should it be very similar? Or can it be completely different? That's an open question. And now this comes to the point that Frank, was bringing up, in the beginning. You click once more, I showed only one transition. There could have been many transitions on the early Earth where conditions were changing rapidly. Meteorites hitting, temperatures changing vastly, catastrophic, events taking place, continent, this, that, those kind of land masses appearing, disappearing, those kind of things. So the question now is, if we run the same chemistry and we catch it in a different time, can we go to something alternative life? Could there have been, if the conditions are not what it is today, of water as the solvent, a pH close to neutral, with the type of atmospheric gases, on with the type of temperatures that we have, if you change any one of those, then the result would be different, because we know chemistry and the chemical laws and physical laws are universal, but they very much depend on the conditions under which you apply to a molecular reaction. So click once more, you will get, we can end up with different biological paradigms. We need not have the same type of life based on DNA, RNA, and proteins. It could be different. It could be deoxypeptide based chemistry, for all you know. I'm giving that in jest as well. So click once more, and that goes to the central point of this lecture. The molecules that we have in today is a destination, not a destiny. It's a destination. If you change the railway track, your destination will change. You change the molecules, you change the conditions, your destination will change. That means when we are looking for life in an exoplanet, we are biased by the single data point that we have in the universe of our own life, and looking for only molecules based on our life will be the same mistake that we make of trying to look back into prebiotic chemistry and looking only for molecules that biology uses today. And click once more, you go to the very last slide, which says what and how can we know about the chemical origins of life. And here I wax a little philosophy. Click once more. This is from an ancient text, from the East, which says, it is known to whom it is unknown. To whom it is known, he knows not. To those who know, it is unknown. To those who do not know, it is known. And I'm sure that you people are saying Ram has lost his marbles. Working in this field, he has gone goo or gaga. But I will leave you with another quote from a very famous practitioner in this field, who said it in a more scientific way. Anybody who thinks they know the solution to the problems of origins of life is deluded. But anybody who thinks it's an insoluble problem is also deluded. So on that note of delusion, I'll stop my lecture and take questions.
Wow, thank you so much for this really wonderful presentation and taking us through all the steps and all the different experiments and reactions you found. And this was really such an amazing talk, and finishing with that more philosophical question is really a great discussion start, I think. So thank you. This was wonderful. And everyone, if you have questions, flash your microphone or post them in the chat. Let's start with the people that are on stage, and then I'll read the questions that are in the chat. Thank you. Yeah, Frank, you had the
Yeah.
question. Yeah, go ahead.
I just want to second you that this is Ram. This is again a great intellectual treat. That is kudo, and thank you to your preparation of this beautiful slides. By the way, I so far logistically I understand you put the slides in viewing only mode. Is it maybe I shall request after this? I mean, it would be great to have have a PDF or some sort of alternative version to keep as a record. This is a lot of things to digest, by the way.
Yeah, I will try to do that. I have to ask my Scripps people, because we are a private research institute. They are sensitive about sharing such information. But these are all in the public domain, so it should not be a problem, because all of this is published work. I don't see any issues with that, but I will get back to you on that. I generally check, because we are a private university and not a the public type one. But I will check, and I don't see any problems, but I usually check to ensure that there is no problem with the IT department. The intellectual transfer, property transfer one. That's it. But I will do that, definitely.
Terrific, thank you. I'll probably message you later. So, by the way, just so for one question for now, and I'll share the mics to other audience. Um, without referring specifically to a particular slides, just recall from memory that you showed somewhere that there are alternative. I remember 3 cycles to all leads to the same. Somehow they also functionally connected to each other, or somehow like to in the end to produce the biology like end products. And so I guess I guess
Do you
still
want to
trying
go
to
to that particular slide? If you give me the slide number, I can go to that if that makes the conversation easier for you.
well, okay, let me do that. Oh, by the way, just
Is it towards the
as
end?
I'm
Yeah, go
yes,
ahead.
as I'm backtracking. So another question on the for the slides 25th and 25. So
Yeah,
your message, one of the messages, the chemical evolution. So could you just by these color of dots, first
yeah,
it's very atom-like, and then they chain up, and then somehow they form sort of a peptides type of a thing. Then there's the end result, purple. Could
yeah.
you give names or a specific like
Sure, sure.
example?
So
Just
they could be peptides like what I showed you before. You start with mixture of hydroxy acids and amino acids will be the different colored balls, and they can combine in different ways to make the peptides of various combinations, which would be the second colored heterogeneous, um, and then as you proceed further, the heterogeneity will slowly decrease and you move to a more homogeneous system, and that is what the all purple color is showing. We have also shown this for nucleotides. For example, I can start with mixtures of RNA and DNA, and we have shown that actually the mixtures help RNA and DNA replicate better than the pure RNA and DNA themselves. If you try to replicate pure RNA and DNA, the problem is the pure RNA binds to itself very strongly, or the pure DNA binds to itself very strongly, or the pure RNA and pure DNA bind to themselves very strongly, that it prevents further replication once it forms the other strand. But if you use these mixtures, what we call chimeras, chimeric strands, they actually help the replication of the pure strands better, and they overcome what is known as a template strand inhibition, and they produce more of that heterogeneous, homogeneous purple color length oligomer, much better than the pure ones themselves. So that's what we are trying to show here. It's like the mixtures provide actually a much better solution of how things can move towards a homogeneity. So what I call this on the left hand side, I call it as a heterogeneous heterogeneity. On the right hand side, I call it a heterogeneous homogeneity, because if you take RNA, it's homogeneous in its backbone. Peptides, they are homogeneous in their peptide nature. And if you take lipids, they are homogeneous in the terms of having a phospholipid structure, okay, throughout. And these all come together. So you have homogeneous systems that come together and work together in a heterogeneous sense, but they themselves are not that heterogeneous. They are homogeneous. So the question is, how do I go from a heterogeneous heterogeneity to a homogeneous heterogeneity? And that is for me the essence of, or the philosophical thrust of our research program, which is captured by these colored balls going from a mixture of heterogeneous colors towards a homogeneous colored oligomer, but it doesn't function alone. It functions only in the presence of others.
so environment must be play a big role in the answer.
Oh, absolutely, absolutely. And that is why I had that last slide where we were going to, if you go to slide number, I think 24, I think, right? Yeah. Let's see, is it 24? No, sorry, it should be slide number 26. Yeah, 26. And if you do the animations there, you will see how you can have, at various stages of change, or switch over, if the conditions changed, then it will change. For example, I'll give you a concrete example. We think about RNA and DNA base pairing and we take it for granted, right? But that is only at a pH 7. You change the pH to a 4, and if the ocean was acidic at always pH 4, none of your Watson-Crick canonical bases would work because they will not base pair. Or if your C was always alkaline around pH 9.5, your base pairs would not form. But then you may come back with a question, but there are life forms that are living in too acidic conditions or in very basic conditions. That is an adaptation that life has come about. If you look at how they do it, they either pump out the protons in an acidic conditions to keep the cytoplasmic pH around 7, or they keep the protons inside at a very basic pH so that they don't lose it, and thus they maintain the pH balance somehow for that Watson-Crick base pairing to work. If your pH was not 6.5 to 7 physiological pH, your RNA bases will never pair. They will fall apart. And that is the principle behind denaturation of RNA and DNA when you put it in acid or on high basic conditions. So environment is absolutely crucial.
That's great illustration example. So I found the slides that I originally wanted to. It's actually 24.
Okay.
If you go yeah. So in the middle, the color red, like comment, you said the combination of all three of them, the cyanide, the ammonia, the keto acids, so produce they all together, right? So that's what you mean.
Yes, that's correct.
Produce
that is a mixture. Yep, that's right.
The next generation, say, amino acids, and the
pyrimidine nucleobase precursors,
pyrimidine
DHA, and those two are the DHA and orotate, the dihydroorotic acid and orotic acid themselves.
So for actually listening to this lecture, I got the perplexing question that I was thinking something different from this comments, this facts that you found. So for me, there, of course, somehow I didn't completely understand. You proved there's only one way to get the pyrimidine, but so for me, yeah. So could you somehow like in this in a sentence or two, like restate that your proof why there's only one that you showed?
Not only one way. What I just said was that there is a chemical reason why the pyrimidines are the only product coming from the reverse citric acid cycle. So if you go and look in biology at the reverse citric acid cycle or the citric acid cycle, they produce the various amino acids. So they produce aspartate, they produce glycine, and they produce alanine, and they produce the glutamate from the 4 keto acids that are there. And then if you look at the nucleobase that biology produces, it's only the pyrimidine orotate. It does not produce uracil, it does not produce cytidine, cytosine, it does not produce adenine nor guanine or thymidine. Those all come later. What I'm trying to say is we found a chemical reason why orotate should be the only one that should be. It's not a biological reason. That's what I was trying to say. So I hope that clarifies it for you, that what I was trying to say was the fact that we find orotate naturally emerging from these reactions tells us that there is a chemical reason why orotate is the only one and why you cannot produce the other nucleobases from this type of chemistry that I've shown. There might be other type of chemistries, but this type of chemistry naturally produces only the orotate, which seems to be coincidental with what biology is doing today.
Gotcha. So this is actually the intermediate product of oxidate. Okay. So that actually leads to my question that I want to ask. So I got the conjecture, or is it there's something actually a theorem type of already established in this field of chemistry answering the origin of life. So the more complex the emergent form could be achieved by multiple ways. So there's some sort of robustness against the changing of the environment or some sort. For example, your cyanide the pathway or the cycle could be you use the word prebiotic, but if we go back to that age of time, Earth, it could be that was the dominant biology, but somehow due to some sort of change of environment, it was become biological, right? So what I'm trying to allude to is that could it do, what do you think? What's your comment on what so is it provable that the current metastable form of our current biology is actually, there's a multiple pathway, so that provides that the some sort of robustness against change of
Yeah.
environment?
That's a very difficult question to answer, because you are asking me to go to a time and hypothesize where I have no data to hypothesize on. Remember, as I told you, go to the black boxes from the very first few slides I showed you. I can demonstrate all these things, but the question will still remain: did this happen on early Earth? Could this happen on early Earth? For which I have no certain answer. The same would be true for people who would come up and ask, can there be life forms based on the cyanide chemistry that you showed us? No way of knowing for sure. I can say yes, probably, but how do you know that's not adaptation of life? If you find some life form that is living on cyanide, can I make a claim that oh look, I found a life form on cyanide, therefore my chemistry is validated? I do not know. It could be an adaptation, and I don't want to confuse adaptation with origination. Okay, that is what many people do. That is exactly the problem with the thermal hydro vent people. They are confusing adaptation of life at thermal vents with origination of life at thermal vents. Those two are not the same. They are completely different. So one has to be very, very careful in trying to make too grandiose of claims. And so that is why I leave that question open all the time. Maybe possible, but we do not know.
That's fair. I think I get the message. That's yeah. So I'll give the mic back to Katerina. I think there's audience that probably if I mean for the audience and also interested in asking question, please do raise your hands so that we can invite you up. Katerina.
Yeah, thank you. Yeah, you asked a question I wanted to ask too. Um, Frank, your last question was, the if there were probably many origins of life on this planet, and then a few, maybe one basically, and were stable enough. But yeah, my question also was if you think that there were many different origins of life on the planet, and then we got, through adaptation, and what survived and was stable enough to the current versions we have.
Yeah, that's entirely possible. We need not even go that far above prebiotic chemistry. Even the Luca, the last universal common ancestor, there are arguments about whether there is a Luca or Lucas. Not the George Lucas, but Lucas. Are there more than one common, universal ancestor? And there are people who argue for that. And the myth of a single common, Luca has been shaken quite a lot by the recent. So
Yeah, thank you. But yeah, I always think it would be so fragile to just rely on one thing, single occurrence. Most of
correct.
things that
Yeah,
occur one time don't make it for a long time.
yeah, absolutely.
Yeah, we had a few questions
Please.
in the chat. So Jake, he asked if there is some encyclopedia or like a place where these cycles are described, where people can look it up. And
Yeah, the cyanide, the cyanide interaction in biology is known from more of a fatal viewpoint, where, what the toxicity of cyanide is. So if you Google, you will get a lot of reactions. And interestingly, you will find that cyanide interferes with the metabolism cycles that I just showed you. For the same reason, it forms cyanohydrin adduct and interferes. So there are many people who have tried to even treat cyanide toxicity using glyoxylate that I showed you, because it can make a glyoxylate adduct and you can pull it out. So from a biological viewpoint, the only thing that I know is cyanide toxicity that has been studied. From a chemical viewpoint, we are the first ones to look into the cyanide chemistry in this kind of a systematic manner. But if you want to look at cyanide chemistry per se in prebiotic chemistry, it's a huge, there are plenty of papers by one of the pioneer in this field is Clifford Matthews. He published a science paper talking about cyanide and hydrogen cyanide polymers at the origins of life. So there's, we have written an accounts of chemical review where we have talked about the cyanide chemistry, where we have tried to kind of collate all the cyanide chemistry. So I can send Katrina the relevant literature and she can distribute it to people who are interested in looking at the cyanide chemistries that are available.
Yeah, thank you. That's wonderful. And the other question Jake had, which you kind of, I think, already answered throughout the talk, if people are in the lab trying to mimic different environmental properties to replicate what happened during that time on Earth.
That's a very valid question. And many people ask me, you have not used any metals, you have not used any clays. Who were your reactors? And I always tell my, students, there were no sigma companies selling you reaction vessels like round bottom flasks or chemicals, which we do as a matter of fact to demonstrate this chemistry. So we have to rely on the minerals, we have to rely on prebiotic reactors, right, if you will. But the problem is, if we go that route immediately, you get a lot of mess, because these are things that go run in different directions based on what the compositions of the minerals are, and many of the analysis become intractable. And one of the important things that my professor taught me was that if you do this research, do the research in such a way that even if you remove the prebiotic relevance, your science stands on its own as a contribution to chemistry or biochemistry. Because that's the only way the poor graduate students and postdocs who work on this project will get jobs, right? Otherwise, they will, you always make a mess and then you publish, oh, I made a mess. Nobody's going to hire that postdoc or student. So I need to be very, very careful. And that is why we select projects this way, to show the proof of principle. And then there are many other colleagues of mine or collaborators of mine who are willing to do this because they are well established and they use their, fancy analytical tools and technology to see, what happens if you add minerals to this. What happens if you add divalent metals that were, plenty present in the ancient ocean or things like that. So yes, I'm all for it. But at this moment, right now, we are not willing to step into that because of the complexity. And I'll give you one concrete example, really concrete example to drive home the point. In 1953, two major papers appeared and changed the facet of science for the whole world. One was the very famous Stanley Miller paper, the Urey Miller, gas spark discharge experiment, which showed you can make amino acids from gases. The second was the Watson-Crick paper, which showed the DNA structure. Okay, now ask yourself, which paper has really stuck in your mind when you think of a game-changing, paradigm-changing chemistry? It's the Watson-Crick that stands out. People talk about it all the time. Stanley Miller paper is only talked about in the context of origins of life as a very pioneering experiment, but even that is being questioned because the reducing conditions are being challenged, right? So what that is always on the back of our minds when we ever fashion projects inspired by the questions of origins of life, what sort of chemistry projects we can fashion that can stand out on its own. In our case here that I showed you with the metabolism, the use of cyanide as a reductant, it can reduce carbon oxygen double bonds, carbon double bonds, has never been shown before. So for me, even if you take the prebiotic relevance, it's a paper that stands on its own by chemistry. And those are the type of projects that we try to do. So Jake's question is completely valid. I have no arguments against it. It's just that for practical purposes, And for people's lives who hang on it to get a job, I have to be careful as to how far I can push this.
Interesting. It's so interesting to learn the thought process, how to approach these in different fields. I find it fascinating. So thank you for sharing that, because we have very different ways of approaching problems, and it's always so much valuable to learn from that.
Yeah.
And Ethan, he's asking if you could comment on Nick Lane's ideas. I don't know,
Oh yeah,
probably
yeah,
you.
Nick Lane, that's correct.
Yeah.
He's a fantastic scientist. I have great admiration for him. He comes from a very different angle. I have taken issues with the conclusions he comes with, because he is trying to, as I told you, try to reconstruct biology based on what metabolism is doing today. So, for example, he will take carbon dioxide and do a reduction, with metals, and he will produce formate, or he'll produce acetate, traces of pyruvate, and then make claims that he has solved the problem. But that's not, for me, that's not that. What you've done is produce the same type of chemistry that you find on a meteorite, right? But if you take a meteorite and put it in a pond, you're not going to get life crawling out of it immediately, at least. Nobody has done it, or have done it and failed. So just by production of these building blocks, you can't claim you have made life. you know for example, you can make pyruvate, but what good is that pyruvate if it does not do anything further under the conditions you have produced? If there is more decomposition than accumulation, you are actually defeating it in a way that you are not in a self-sustainable mode. You're not in a, leave alone self-sustaining, not in a sustainable mode. But I have enormous respect for the chemistry that he demonstrates, and the gust, the conviction with which he does. All more, more power to him, because we need people coming from different directions to convince each other through actual experimental results rather than just claims.
Yeah, thank you so much for your answers. And Denise and Kirko, did you have some more questions? No, I know we've been going on for almost 2 hours, so it's the last few minutes, and then I would like to give Dr. Ram a break. Thank you.
No, but there are plenty of good questions. There are focus on cyanide pathways that explore formaldehyde and chemical agents. Yes, formaldehyde has been the very famous Formose reaction I showed in the very beginning of my slide. That Formose reaction uses formaldehyde as the source molecule to make sugars. People have been using it. And so there are a lot of chemistries here. As I said, I've only shown you a facet of chemistry based on my philosophical approach. Doesn't mean that's the only one or the right way to think about it. Different ways to think about it, and people have come up with great discoveries because of that. And so we all need to kind of look at the different approaches and see what sort of ideas emerge that become naturally a front runner for people to take on, like the RNA world theory. Many people still hang their hats on an RNA world theory. I don't belong to any world theory. I just belong to I want to do these reactions and let the system teach me how it moves, rather than me teaching the system how it moves.
I did not have any questions because everybody had asked them. I just wanted to say that the slides were exceedingly excellent, and appreciate you for that and taking us through the entire process. This was an amazing way to spend this evening. Thank you so much.
Thank you, thank you, thank you so much. I will try to get the PowerPoint slides in the animation so that you can follow that, and I will talk to my the people and find out if there is any problem sharing it. That's all.
Yeah, thank you so much, and thank you especially also for the answers you gave us. Frank, you wanted to
Yeah,
to
if I may, I just want to add one more question. I mean,
Yeah.
okay, yeah. So so for the in your I would say a alternative route achieving the biology. Where I do see in the slide somewhere the role that the clay is it in the hydration the part of it, or so in your setup or model that has been realized, how important the wet and dry cycle, how many roles, critical functions that that particular environment provides versus other.
You mean the cycling, wet dry cycling? How many cycles? Was that the question? I'm sorry, I didn't understand that.
Right. So, so in yeah. So the question, if I don't think I have is myself very clear, but the so what in your demonstration, which is different from the hypothetically the real, what might have been going on in real biology. So let's just give it as may be. So let's say I'm believer of when dry cycle,
Yeah.
assumptions, but then what your model, if I'm looking for supporting or pro and con, or unfavorite facts from your model, what's your model can have to say about when dry cycle?
So wet and dry cycles do have a role to play. They certainly do. But what I'm trying to move is towards chemistries that can happen in solution so that I can do it within an enclosure like a protocell. Okay. So once you go into the protocell, the need for wet and dry chemistry goes down. So the idea for me is the wet and dry cycle will give you certain molecules to start off with, but then if those molecules can become independent of that wet dry cycle, those are the molecules that could go further. If you are going to be always dependent on wet dry cycle for your survival, formation and survival and existence, then you will be very limited. You see that? So what I'm trying to find out, I'll give you a concrete example. For example, going back to the deoxypeptide to peptide, which is only possible via the peptide, wet dry cycle. Let us suppose this is a Gedanken experiment, a completely thought experiment. Let us suppose you come up with a deoxypeptide or a long peptide or a deoxypeptide which is able to make peptide bonds. That's not unheard of. Biology uses peptides to catalyze peptide bonds. There is a non-ribosomal peptide synthesis. So if there is a peptide that could begin to make peptide bonds, wouldn't that peptide start making peptide bonds without the need for wet dry cycle? That is what I'm thinking. That's the way I want to move. I'm not against wet dry cycle, but the wet dry cycle is not the solution from start to finish. It plays a role, it has its part, but it will be the 18th camel which goes away after playing its part. Something else will take its place. And that's the nature of evolution. Something always better always takes the place of the older one, right? And so wet dry cycle has an evolutionary part and role to play, but it might go away when something else comes along which is much better than that.
I see. So that's again, thank you. That's probably quite crucial message from your sharing. That's interesting.
Yeah.
Great.
I think if you still have time for one more question, John.
Please, yes. Yeah.
John? Yeah, thank you. Hey, John, go ahead.
Yeah, hi, Katerina. Thank you for giving me the chance to ask the last question. My question is, as we know that in our protein, all the amino acids are L type, and our glucose pretty much all are L type. So does that mean anything about the origin of this our life? I'm thinking about if we have a mixture of all those non in organic molecules, and then they make organic molecule. Most of the time, they probably will make see different types, D, L, D and D type, L type. But see in nature, actually, see what will happen is all the amino acid are L type and glucose L type. So would this be see against the multi-origin theory or just some thought?
Great question. Great question. I didn't touch upon chirality at all for a simple reason. There has been no good explanation as to why it is L amino acids and D, sugars that biology has chosen to work with. Now, the problem is this. Even if you do find a way of making pure amino acids, L amino acids, and pure D sugars, you will not be able to keep that chirality for long because they will get destroyed. And you might say, how wrong? No, but think about it. Look in biology, what biology does for the transamination. If you go back to the slide of transamination, you will see in the transamination that chirality is destroyed because that amino acid becomes a keto acid, right? So, but biology solves it beautifully by using enzymes which impose that correct chirality. So if you do that transamination without enzymes, your amino acids would racemize completely. And the same is true for sugars, because the sugars have an aldehyde, and and many heating processes, or heating with metals and things like that, they can scramble the stereochemistry. In fact, we have submitted a paper where we show that the carbonyl group can actually walk itself across the backbone of the sugar and scramble the stereochemistry. Okay, so my problem is not trying to understand why it is L and D. That is mind-blowing and beyond my ability to comprehend or explain. But my problem is more tractable. Why a homochiral oligomer? Why is it completely of L, and why it is completely D in the RNA?
Yeah.
And that is simply because it's only the homochiral polymer that can function. So for example, if you take replication, it's only the homochiral polymer that replicates better than the heterochiral polymer. The same way, if you take a homo L amino acid peptide, that resists hydrolysis much better than the chiral mixed ones. So I can tell you whether it can be completely homo L or homo D, but I can't tell you why it is D or L. That choice comes at the level of a polymer, not at the level of a monomer. And that is my submission for people who worry about chirality, is that the chirality at the small molecule level is very difficult to control. But the chirality at the level of an oligomer is much more easier to control because it's selected based on a function. So if I have a mixture of D and L mixed oligomer versus a pure D oligomer or a pure L oligomer, it's the pure D and L oligomer which will move forward based on their function. The mixed one will suffer quite a lot of problems. And I can begin to see why you can have this, what I call this homogeneity in the biological molecules, because they function better. And biology relies on function for survival, and therefore it chooses a homo L or a homo D. But why it is D or L, that is not known. And I do not know whether we can find it to any satisfaction unless a great discovery is made.
Yeah, I'm just wondering, see, if in another world, see, another Earth,
It
maybe
could be the
see
other way
all the
around.
amino acid. Yeah.
Actually, you should read Alice in Wonderland. There she wonders about the mirror images of the milk molecule. So it's an interesting idea. And actually, there are some people who have scientists who have synthesized the completely D version of a protein, and show that it works exactly the opposite way of the complete L version of the protein. So it works on the opposite chirality of the substrate. So they have shown it. So it is possible. It is possible that you have a life that is completely based on D amino acids and L sugars.
Thank you.
Actually, my colleague Donna Blackman at the Scripps Research Institute is one of the foremost in trying to tackle this chirality question in terms of origins of life.
Yeah, so that's a quite interesting discussion that just heard over there. Thank you. The I have a more basic question on the slides number 6. You showed a. Oh, by the way, on the slides, there's an interesting quote that you shared. Earlier, I took it wrong. I thought you said evolution is a thinker, not an engineer. But now I see it's actually tinkerer.
Yeah, tinkerer.
I got it
Oh, no.
exactly opposite. Anyway,
slide number 6.
Yeah, slide number 6. So at the lower, on the right lower bottom, there you see the ammonium cyanide.
Correct.
Yeah. So they go up like there are 3 candidates that they can form be in.
They do
so
form. It's not a can form. They do form that. So that means the ammonium cyanide makes a polymer, and the dark polymer, when you hydrolyze, these molecules come out.
Right. So what is the background knowledge of this for the only the middle ones goes up, so
No,
goes into
so
the real
the
body?
idea is the idea there was that that people are looking at RNA, which is at the very top, and they are looking at the molecules uridine and cytidine, and they are looking at the nucleobases uracil and cytosine, and they see the same molecules forming in the ammonium cyanide reaction, and they make an immediate connection saying that, look, these molecules are made, therefore life should make RNA. But my question is, if you look how biology makes that RNA molecule, it does not make it from uracil and cytosine. It makes it from orotate, which is right below. And that orotate comes from orotic acid. And the thing is, this orotic acid is also found in that mixture coming from ammonium cyanide. But because people are biased by RNA and what they see in RNA, they don't pay any attention to the orotic acid that is in this mixture. And my point is that if you want to look into biology and how biology makes it, why don't you study orotic acid? Why are you only looking at uracil and cytosine? That is the point that I was trying to make.
Okay. No, got it. Okay.
Well, thank you so much. Thank you, John, for coming up and asking that question. That was a very interesting discussion also. And thank you, everyone, for asking great questions. And of course, a special thank you to you, Ram. This was such a treat to get to learn so much from your field and dive into biochemistry again, which I haven't been doing for a while. And I learned so much, and I think everyone here, you can see in the chat, and people reaching out saying thank you so much for this room, that this was an amazing talk. So yeah, thank you, and we really appreciate it, and feel free to come back anytime again when you maybe want to share some research updates. We are always very happy to welcome you back. So thank you so much. This was amazing.
Yeah. Okay. Thank you so much for this opportunity, and thank you all for your kind attention and questions. Yeah. Thank you. Bye.
Yeah, thank you. And everyone, if you like discussions like this, just follow the club. Then you get updates and you see the calendar.
Yeah.
Tomorrow we have 2 rooms because we had to reschedule previous room. So it kind of ended up having 2 rooms tomorrow. So Dr. Fradkin from Israel will join us to talk about methods how to control unwanted thoughts. And Dr. Lana will talk about intracellular transfer of telomeres in T cells. He's a aging scientist for rejuvenation. He also has a company that he formed recently with a really interesting approach on aging and how to treat aging. So yeah, involving T cells. So yeah, come back. I hope I hear you all back soon, and have a great rest of the evening, day, or Frank, rest of your day. I think it's morning where you are. So thank you, everyone. And again, thank you so much, Dr. Krishnamurthy.
Yeah,
And
thanks again. Yeah, thanks
you
again.
will
Very
hear.
grateful for this opportunity. Yeah,
Yeah, thank you. Okay,
yeah.
close the room and 3, 2, 1. Bye, everyone. Thank you.
Thank you, Katerina.